In the heart of West Africa lies Burkina Faso, a country shaped by savanna, Sahel, ancient trade routes, powerful kingdoms, pastoral migrations, farming communities, and generations of human movement. On a modern map, its borders look clear. But DNA does not follow modern borders. Inside Burkina Faso live many communities with different languages, histories, and ancestral connections. Scientists studying their DNA have found something remarkable. Maternal lineages and paternal lineages do not always tell the same story. Some genetic patterns follow geography. Others appear connected with language, marriage traditions, migration, pathogens, or pastoral life. Among Fulani communities, even the ability to digest milk in adulthood preserves clues about ancient population contact. Among other populations, malaria has left its own evolutionary pressure. This is not a story about racial purity or one national genetic profile. It is an investigation into movement, adaptation, relationships, and human resilience. To understand the DNA of Burkina Faso's people, we must first understand why there is no single Burkina Faso DNA. When people hear the phrase DNA of Burkina Faso, it is easy to imagine that scientists could collect a few samples, calculate several ancestry percentages, and produce one genetic profile for the whole country. But population genetics does not work that way, especially in a region with as much cultural and historical diversity as West Africa. Burkina Faso is a modern nation, while the ancestry carried by its people began forming thousands of years before its modern borders existed. The country includes many ethno-linguistic communities, including Mossi, Fulani, Baisa, Samo, Marka, Kassina, Nuna, Laila, Pana, Samoya, and others. These communities do not all share the same history, language, social organization, or migration experience. Some languages belong to Gur branches, others belong to Mande branches, while Fulfulde belongs to another branch within the wider Niger-Congo language family. This means several different maps exist across the same territory. There is the political map of Burkina Faso. There is the linguistic map. There are maps of old kingdoms and migration routes. There are ecological zones stretching from the Sahel toward wetter savannas. And underneath all of them is the genetic map. Scientists have tried to understand how these maps overlap. One important study examined hundreds of individuals belonging to 10 ethno-linguistic populations in Burkina Faso. Researchers were interested in whether language, geography, or social traditions had left detectable signatures in the DNA. They analyzed two particularly revealing forms of genetic evidence. One was mitochondrial DNA, which is inherited mainly through the maternal line. The other was the Y chromosome, which is passed from father to son. These two genetic systems represent only small parts of a person's complete ancestry, but because they travel through families in different ways, they can preserve different demographic histories. The results immediately showed why the idea of one Burkina Faso genetic profile is misleading. The maternal DNA of the sampled populations was highly diverse but relatively similar across many ethno-linguistic boundaries. The paternal DNA showed stronger divisions. In particular, some Y chromosome patterns appeared more closely connected with linguistic affiliation. This contrast suggests that men and women did not always move between communities in the same way. In many of the populations studied, marriage traditions historically involved women moving to the husband's community. Men were more likely to remain near their paternal relatives. Over many generations, this can create a very specific genetic effect. Mitochondrial lineages move between communities because women carry them into new families. Y chromosomes remain more strongly associated with local male groups. Eventually, two neighboring populations may speak different languages and retain different paternal patterns while sharing many maternal lineages. This is a powerful reminder that genetic structure does not always come from ancient isolation. Sometimes it comes from ordinary social behavior repeated generation after generation. Marriage can reshape genetic geography. Residence patterns can reshape genetic geography. Political expansion can reshape genetic geography. Trade, territorial struggles, migration, and the incorporation of outsiders can do the same. Later research examining more male samples from many ethnolinguistic populations across Burkina Faso added another layer. It found extremely high Y chromosome diversity and showed that geographic proximity was also important. Neighboring populations often appeared genetically closer than distant populations. This tells us that language was never an absolute barrier. People interacted. Men moved too, even if their movements sometimes followed different patterns from women. Communities that maintained distinct identities still exchanged genes with neighbors. That is how real human history usually works. Populations are not sealed containers. They are networks. Some connections are strong. Others are weak. Some continue for centuries, while others emerge after migration, territorial shifts, marriage, or economic change. Burkina Faso sits at the meeting point of several historical worlds. To the north lies the wider Sahel, a zone crossed by pastoralists, traders, and religious networks. To the south and west lie regions connected through farming, trade, and Mande-speaking populations. In the central regions, Mossi political systems became influential. These processes did not replace one another neatly. They overlapped. A person today may carry ancestry created by many of them. This is why responsible population genetics begins not with the question, what race are these people, but with more precise questions. Which community was sampled? Where were the participants living? Which genetic markers were examined? Were researchers studying maternal ancestry, paternal ancestry, or the entire genome? What comparison populations were used? How large was the sample? And what historical explanation best fits the data? Once those questions are asked, Burkina Faso becomes far more interesting than a simple ancestry percentage. Its DNA becomes evidence of relationships. Maternal lineages reveal movement across social boundaries. Paternal lineages preserve other kinds of structure. Whole genomes reveal wider regional connections. Genetic adaptations record encounters with diet and environment. The country is therefore not a single biological unit. It is a crossroads where different population histories have repeatedly met. And the first of those histories becomes especially visible when we follow the genetic line that passes from mothers to their children. Every person carries mitochondrial DNA, a small genetic archive located inside the mitochondria that help power our cells. Unlike most of the genome, which is reshuffled when DNA from two parents combines, mitochondrial DNA is inherited almost entirely from the mother. A mother passes it to both sons and daughters, but daughters are generally the ones who pass it into the next generation. Because of this inheritance pattern, scientists can follow maternal lineages far into the past. Over time, mutations appear in mitochondrial DNA, creating branches that geneticists call haplogroups. These haplogroups can help researchers reconstruct ancient population relationships, but they must be interpreted carefully. A mitochondrial haplogroup does not describe a person's entire ancestry. It represents only one maternal line among the enormous number of ancestors that contributed to that individual. Nor does a haplogroup equal an ethnicity, tribe, nationality, or race. The same ancient maternal lineage may be found among many populations whose languages and identities are very different today. When researchers studied mitochondrial DNA from multiple ethnolinguistic groups in Burkina Faso, they found extremely high diversity within the populations. Several lineages belonging to major African branches appeared repeatedly, including different forms of L1, L2, and L3. One especially widespread branch, L2A, occurred frequently across the samples. Yet the most important discovery was not that one haplogroup was common. It was that the maternal lineages did not divide neatly according to language. Gur-speaking and Mande-speaking populations could show considerable overlap in their mitochondrial ancestry. This was very different from what researchers observed in the Y chromosome. To understand why, imagine a woman born into one linguistic community several centuries ago. She marries a man from a neighboring group and moves to his settlement. Her children grow up surrounded by their father's relatives. They may speak their father's community language and identify socially with that community. Yet they still carry their mother's mitochondrial DNA. Her daughters pass the same maternal lineage to their own children. If this process happens repeatedly across hundreds of families and many generations, maternal DNA begins crossing linguistic and cultural boundaries even when the identities of those communities remain distinct. This is one reason the mitochondrial landscape of Burkina Faso appears relatively shared across groups that are otherwise different. Researchers connected this pattern with traditions of patrilocal residence and marriage between communities. Women often moved to the husband's place of residence, while men were more likely to remain closer to their paternal community. DNA therefore preserves evidence of social organization. The movements may have been small, perhaps only between neighboring villages, but multiplied across centuries they become visible in population genetics. The mitochondrial data may also preserve much older demographic signals. Researchers modeling maternal lineages identified evidence consistent with population expansion beginning around the transition into the early Holocene, thousands of years before any modern Burkina Faso ethnic identity existed. This was a period when African environments were changing dramatically. The Sahara, now one of the driest regions on Earth, experienced a much wetter phase. Lakes expanded. Rivers and wetlands appeared in areas that are now desert. Grasslands stretched across large territories, creating new environments for humans and animals. These ecological changes may have encouraged population growth and opened routes of movement between regions that later became separated by severe aridity. But a genetic signal from that period cannot be assigned directly to modern Mossi, Fulani, Bisa, Samo, or any other present-day group. Modern ethnic identities formed much later. The ancestral populations living thousands of years ago would have had their own cultures and identities, many of which are unknown to us. Genetics reveals demographic patterns, not the names people used for themselves. Archaeology therefore becomes essential. Burkina Faso contains evidence of deep technological development, including ancient ironworking traditions. The archaeological remains at Durula show iron production dating to the 1st millennium BC, demonstrating that communities in the region were developing specialized technologies long before many later political formations emerged. These archaeological sites remind us that ancestry is not the whole story of civilization. A community can adopt a technology without being replaced genetically. A language can spread without every person carrying the same ancestry. A migrating group can settle among existing populations and become part of a new society. When we combine mitochondrial evidence with archaeology, the story becomes more realistic. The maternal ancestry of modern Burkina Faso populations reflects thousands of years of population movement, but that movement did not erase local cultural development. Instead, people mixed while communities continued to create technologies, traditions, political systems, and languages. This is why no group can claim ownership of an ancient haplogroup simply because it appears frequently among its members today. Haplogroups are older than modern ethnic categories. Their value lies in showing connections across time. In Burkina Faso, the maternal map tells us that women repeatedly crossed boundaries. Their movements connected populations that languages sometimes kept separate. Their descendants carried these lineages into new communities, generation after generation. Yet when scientists turned from mitochondrial DNA to the Y chromosome, the picture changed. The paternal map contained stronger boundaries, unusual founder effects, and clues that some male lineages expanded dramatically during particular episodes of history.
The Y chromosome tells a different kind of story because most of it passes directly from father to son. Only males normally carry a Y chromosome, and although it represents just one line within a person's ancestry, it can preserve distinctive signals of male population history. If a small number of men establish a new community, gain social power, survive a population decline, or have unusually large numbers of descendants, their Y chromosomes can become extremely common. Centuries later, that pattern can remain visible even though the rest of the genome has mixed extensively with neighboring populations. When researchers examined the paternal lineages of ethno-linguistic groups in Burkina Faso, several branches of Y chromosome haplogroup E were frequent. This broad haplogroup is widespread across Africa and contains many subbranches with different histories. The interesting result was not simply that haplogroup E appeared. It was that the frequencies and Y chromosome patterns varied substantially between sample populations. Compared with mitochondrial DNA, the paternal data showed stronger structuring according to linguistic affiliation. Some Gur-speaking and Mande-speaking communities appeared more clearly separated on the paternal side. This suggested that male ancestry had remained more locally organized while maternal ancestry crossed community boundaries more freely. But not every population followed the same pattern. The Bisa sample provided one of the strongest examples of a possible paternal founder effect. A large majority of sampled Bisa men belonged to one broad paternal lineage, and many shared closely related Y chromosome signatures. Such a pattern can emerge when a relatively small number of paternal ancestors contribute disproportionately to later generations. This might happen after migration, severe population bottlenecks, political expansion, or social practices that concentrate reproductive success among particular male families. Genetics alone cannot prove which event occurred, so researchers must compare the evidence with historical and anthropological information. The Mossi sample also displayed relatively concentrated paternal diversity compared with a wide diversity found in mitochondrial DNA. Other populations, such as the Samoa in the same dataset, showed much greater variation among paternal lineages. These contrasts again show why a national genetic average would hide the most meaningful information. Even populations living inside the same country can preserve very different demographic histories. Later, Y chromosome research expanded the number of individuals and ethno-linguistic communities sampled. One study analyzed hundreds of men belonging to more than 20 groups across Burkina Faso and found extraordinarily high haplotype diversity. In that dataset, the Y chromosome profiles were so diverse that each sampled haplotype could be distinguished. At the same time, geography emerged as an important predictor of genetic similarity. Neighboring populations tended to share more paternal relationships than communities separated by greater distances. This means the paternal map contains several historical layers. Older patterns may reflect linguistic boundaries, founder effects, or long-term social organization. More recent patterns may reflect movement between neighboring communities. Imagine two groups that developed different languages or political identities centuries ago. For generations, most men marry locally, preserving distinct paternal structures. Later, trade expands. Villages become economically connected. Men migrate for work, civic duties, farming, or marriage. Their Y chromosomes begin moving between communities. The older distinction does not disappear instantly. Instead, the new movement is added on top of it. This layering makes genetics difficult to interpret, but also makes it historically rich. One particularly interesting example is the Bisa. Linguistically, they belong to the Mande family, yet some paternal comparisons have placed sampled Bisa men closer to neighboring Gur-speaking populations than to other Mande comparison groups. This does not mean that the Bisa are biologically really Gur, because biological ancestry cannot determine linguistic identity. It means that geography, gene flow, founder effects, or language change may have produced a mismatch between linguistic classification and paternal genetic similarity. Human populations frequently change language while remaining genetically connected to earlier neighbors. A ruling group can introduce a new language. Migrants can adopt the language of the majority. Communities can become bilingual and later shift toward one language. These processes can produce populations whose genes and languages have different histories. Scientists must therefore avoid treating language families as biological categories. The same caution applies to genetic distance. If two populations appear close in Y chromosome data, that does not mean they are identical overall. Y chromosomes represent only paternal lines. Their mitochondrial and autosomal histories may be different. Sample size also matters. A small sample can exaggerate the apparent importance of one lineage. Some Burkina Faso studies contain many individuals from particular communities and only a few from others. That makes broad conclusions about undersampled groups uncertain. Still, the overall pattern is clear enough to reveal a major historical principle. Men and women have not always moved through Burkina Faso in the same ways. Maternal lineages often crossed ethno-linguistic boundaries. Paternal lineages sometimes remained more strongly connected with local populations, language communities, geography, and founder histories. This difference may reflect long-established marriage systems and residence patterns. It may also preserve evidence of political expansions in which particular male lineages became influential.
The Mossi occupy a major place in the history of Burkina Faso. Their kingdoms became some of the most influential political systems of the central West African savanna, and traditions surrounding their formation describe migration, territorial expansion, alliance, and the incorporation of established communities. The genetic evidence does not allow us to recreate every event in those traditions, but it does offer an intriguing demographic pattern. In one major Burkina Faso study, sampled Mossi men showed relatively low Y chromosome haplogroup diversity, while mitochondrial diversity among Mossi participants remained much higher. This combination raises an important possibility. If a comparatively restricted set of male lineages expanded politically or demographically while women from numerous local populations became incorporated into the growing society, later descendants could display concentrated paternal ancestry alongside much more diverse maternal ancestry. The researchers suggested that this pattern was compatible with elements of Mossi expansion history. Compatible is the important word. DNA does not prove that a particular king, battle, or migration described in oral tradition occurred exactly as remembered. What it can reveal is a demographic result consistent with certain kinds of expansion. Imagine a group of men entering a region already occupied by established farming communities. Instead of replacing everyone, they build alliances, establish authority, marry locally, and incorporate existing settlements. Their political identity expands faster than their biological ancestry. Within a few generations, thousands of people may identify with the growing political community even though much of their ancestry comes from populations that were living in the area before the expansion began. If social organization also favors descent through male political lineages, certain Y chromosomes can become especially common. Meanwhile, women from many different maternal backgrounds contribute to the expanding population, maintaining high mitochondrial diversity. This kind of process has occurred repeatedly across human history. Empires, kingdoms, and ruling groups often expand through incorporation rather than total replacement. The resulting genetic pattern can therefore be complex. A population can preserve evidence of a paternal founder effect while its overall genome reflects substantial mixture with local people. This is why Y chromosome results should never be interpreted as the complete ancestry of the Mossi. Every individual carries 22 pairs of autosomes inherited from both parents, and these chromosomes contain contributions from countless ancestors. A Y chromosome follows only one narrow paternal path. Consider a ruler who lived 500 years ago. He might have thousands of genealogical descendants today. Yet only a small fraction would carry his Y chromosome because that chromosome survives only through an unbroken sequence of father-to-son transmission. Many other descendants may carry parts of his autosomal DNA, while others may inherit no identifiable DNA from him at all. A strong paternal signal therefore tells us about lineage expansion, not total population replacement. Modern whole genome data are helping researchers move beyond narrow paternal and maternal markers. Mossi samples have been included in international genomic projects, particularly research connected with malaria and African genetic diversity. These whole genome datasets are valuable for historical population studies, but they are also essential for medicine. African populations have historically been underrepresented in genomic databases, which creates major scientific problems. Genetic variants common in West Africa may be rare in European populations and therefore poorly characterized by medical studies based mostly on European ancestry. Methods designed to predict missing genotypes also perform less accurately when suitable African reference genomes are unavailable. Including Mossi and other Burkina Faso populations improves the quality of both evolutionary and health research. Yet genetics is only one source of evidence for Mossi history. Archaeology demonstrates that central Burkina Faso was not an empty landscape waiting for political organization to arrive. Ancient iron smelting sites show sophisticated technological traditions that stretch back many centuries before the historically documented development of later Mossi political systems. Communities in the region were already producing iron, farming, trading, and organizing labor. This changes the way political expansion should be imagined. Rather than a simple story in which newcomers bring civilization to an empty region, the evidence supports interaction among populations that already possessed complex cultures. Incoming political groups could adopt local practices, recruit local specialists, marry into established families, and incorporate existing settlements. Over time, the difference between newcomer and local could become socially meaningless even if small genetic traces of the original demographic process remained. This is one reason identity cannot be reduced to ancestry. A person does not become less Mossi because some ancestors belonged to communities incorporated centuries ago. Historical societies are constantly created through integration. Political identity can unite people whose deeper genealogies are diverse. Language can spread through institutions and family life. Cultural traditions can be shared by descendants of many ancestral populations. The genetics of the Mossi therefore contributes to a larger lesson about Burkina Faso. Modern populations are not frozen remnants of isolated ancient tribes. They are historical communities formed by movement, alliance, integration, marriage, and social change. The Fulani are one of the most geographically widespread populations of the African Sahel, with communities extending across an enormous region from West Africa toward Central Africa and beyond. Their history is strongly associated with pastoralism, cattle herding, mobility, trade, Islamic scholarship, and later political states, but no single description fits every Fulani community. Some groups maintained highly mobile pastoral lifestyles. Others became settled farmers, merchants, urban populations, religious scholars, or political elites. Their ancestry is equally diverse. This is why a Fulani sample from Burkina Faso cannot automatically represent Fulani populations living in Senegal, Mali, Niger, Cameroon, Chad, or other countries. Nevertheless, genetic research in Burkina Faso has provided one of the most important windows into Fulani population history. Scientists studying pastoralists near Sahara analyzed genome-wide DNA from dozens of individuals. Unlike mitochondrial DNA or the Y chromosome, genome-wide analysis examines hundreds of thousands or millions of genetic markers distributed across the chromosomes inherited from both parents. This allows researchers to detect more complex ancestry patterns. The Sahara Fulani sample showed predominantly West African ancestry, but it also carried a significant component related to populations from North Africa and to ancestry ultimately connected with West Eurasia. In one statistical model, a European-associated component represented roughly 1/5 of the modeled ancestry of that particular sample. This figure is easy to misunderstand. It does not mean that all Fulani are a fixed percentage European. It does not even mean that historical Europeans traveled directly to Burkina Faso and contributed that exact amount of ancestry. Ancestry percentages depend heavily on the reference populations chosen by researchers. If the real historical source population was itself mixed, a computer model may divide that ancestry into several modern categories. Researchers therefore looked beyond percentages and asked how the ancestry entered Fulani populations. Their analysis suggested a more complicated route. The European-related signal often appeared together with North African-related ancestry, supporting a model in which West Eurasian-related ancestry first entered populations in North Africa and ancestry from those populations later contributed to Fulani ancestors. In other words, North Africa likely acted as an intermediary. This makes sense historically because the Sahara has never been a permanent wall. During wetter climatic periods, it contained grasslands and lakes that supported human movement. Even during drier eras, trade, pastoralism, migration, and political networks repeatedly crossed it. The most remarkable clue in the Sahara study, however, was not an ancestry percentage. It was a genetic variant connected with milk digestion. Most humans are born with the ability to digest lactose, the sugar found in milk, because infants depend on milk during early life. In many populations, production of the enzyme lactase decreases after childhood. But some populations evolved genetic variants that allow lactase production to continue into adulthood. This trait is called lactase persistence. It became especially important among populations that relied heavily on cattle, goats, or other dairy-producing animals. Among the sampled Fulani from Burkina Faso, researchers found a lactase persistence variant known as T13910 at a high frequency. This same variant is strongly associated with adult milk digestion in European populations. Scientists then asked whether the mutation had appeared independently among Fulani ancestors or had entered through population mixture. The DNA surrounding the mutation provided an answer. The variant occurred on a genetic background closely related to the background seen in populations carrying the European-associated version of the allele. This strongly supported introduction through admixture rather than an entirely independent mutation. Once it entered the Fulani ancestral population, natural selection appears to have increased its frequency. The reason is straightforward. In a society where cattle and milk are important resources, the ability to digest fresh milk can provide nutritional advantages. Milk contains energy, protein, fat, minerals, and fluid. During seasonal food shortages or difficult environmental conditions, being able to consume it efficiently may improve physical endurance and reproductive success. Over many generations, individuals carrying a useful lactase persistence allele can leave more descendants on average. The allele then becomes much more common than most of the ancestry that originally introduced it. This is a powerful example of culture shaping biological evolution. Pastoralism created the environment in which the genetic variant became advantageous. The gene did not create Fulani culture. Rather, a cultural way of life made a particular genetic trait valuable. Later research using hundreds of Fulani individuals from many populations across the Sahel demonstrated that Fulani genetic diversity is even more complicated. Different communities have mixed with different neighbors as they expanded geographically. Some share more ancestry with populations in western regions. Others reflect greater local admixture farther east. This means there is no single Fulani ancestry percentage or uniform genetic profile. What unites Fulani communities culturally and linguistically does not require biological uniformity. In fact, pastoral mobility may help explain why shared identity exists alongside substantial genetic variation. Mobile communities can preserve language and cultural networks over long distances while exchanging genes with neighboring populations. The Fulani of Burkina Faso therefore provide an especially clear example of how migration, local admixture, and natural selection can operate together.
The story of lactase persistence among Fulani pastoralists is not simply about digestion. It is one of the clearest examples of how human culture can change the direction of biological evolution. For most of mammalian history, drinking milk was something infants did. Young mammals produce the enzyme lactase because it allows them to break down lactose, the main sugar in milk. As they grow and move to other foods, lactase activity usually decreases. Early humans probably follow the same pattern. Then some human populations domesticated animals and began using their milk as food. That cultural innovation created an entirely new nutritional environment. Adults who could continue digesting lactose suddenly gained access to a rich food source that had previously been most useful during infancy. Different populations responded to this opportunity through different genetic routes. Several lactase persistence mutations are known in Africa and Eurasia. Some East African pastoral populations carry variants that are different from the best-known European-associated mutation. This tells scientists that adult milk digestion evolved more than once. Similar cultural pressures produced separate genetic solutions. The Fulani case is especially interesting because one major lactase persistence variant found among them appears to have arrived through admixture rather than evolving independently. In the Zinier Fulani sample from Burkina Faso, the T13910 variant occurred at a high frequency. Physiological testing showed that carriers were more likely to digest lactose efficiently. The genetic regions surrounding the variant also displayed signs of strong natural selection. This combination makes the evidence particularly persuasive. Researchers were not merely looking at a statistical association. They were examining a variant whose biological effect is already well understood and whose surrounding DNA preserve evidence of historical population contact. Imagine the process unfolding over many generations. A population with ancestry connected through North Africa contributes a small amount of DNA to ancestors of the Fulani. Most of the incoming variants have little effect on long-term lineage survival. They are passed forward randomly. Recombination breaks the original ancestry segments into smaller pieces. Some disappear. Others remain at low frequencies. But one piece contains a variant that allows adults to digest fresh milk. In a pastoral community where cattle are central to subsistence, that variant offers a meaningful advantage. Carriers can use milk more effectively as a source of calories and hydration. During shortages, droughts, long movements with herds, or periods when crops are less available, this advantage can matter. Individuals carrying the variant may thrive slightly better or raise more healthy children. The difference does not have to be dramatic in every generation. Even a modest advantage, repeated for centuries, can make an allele much more common. Eventually, the lactase persistence variant reaches a frequency far higher than the overall ancestry contribution from the population that originally introduced it. This is natural selection acting on admixture. It demonstrates why ancestry percentages alone can be misleading. A person may carry a small amount of ancestry from a particular historical source, while carrying one extremely common adaptive variant inherited from that same source. Selection treats useful genes differently from the rest of the genome. This process is sometimes described as adaptive introgression, but genetics tells only half of the story. The reason the allele mattered was cultural. Humans had already domesticated cattle. They had developed knowledge of herding, animal care, seasonal movement, breeding, and milk use. Those practices transformed milk into a major resource. The cultural system came first. The genetic advantage emerged within that system. This relationship is known as gene-culture coevolution. Human behavior changes the environment. The new environment changes, which genetic variants are advantageous. Over time, genetic evolution can feed back into cultural life by making certain foods or practices easier for some populations to use. Yet no single gene makes a person Fulani. Many Fulani individuals do not carry the lactase persistence variant. Some people can tolerate dairy despite lacking the best-known allele, while others may avoid fresh milk or consume processed dairy with less lactose. Fermentation, souring, and other food technologies can reduce lactose and allow dairy consumption without strong biological lactase persistence. Cultural identity does not depend on genotype, nor does pastoralism require one particular mutation. The same caution applies when comparing Fulani populations. A variant common in Burkina Faso may occur at different frequencies elsewhere because Fulani groups experience different histories of migration and mixture. The broader genomic study of Fulani communities across the Sahel shows that shared identity exists alongside considerable genetic diversity. This is exactly what we would expect from a population whose historical networks covered enormous distances. As communities moved, they encountered new neighbors and incorporated new ancestry. Yet some useful genetic traits, especially those linked with pastoral life, could travel widely because natural selection favored them. The milk story therefore captures several forces at once. Migration introduced ancestry. Admixture introduced a particular variant. Culture created the conditions that made the variant valuable. Natural selection increased its frequency. Later population movement carried it into new regions. Few examples show more clearly that human evolution did not end in the distant Stone Age. It continued during the development of pastoral societies and continues today.
For generations, malaria has been one of the strongest biological pressures affecting human populations across much of tropical Africa. The disease is caused by parasites transmitted through mosquitoes, and the most dangerous form, associated with Plasmodium falciparum, has profoundly impacted ancient populations over human history. In regions where malaria remained common for many generations, even small genetic differences affecting infection or severity could influence which variants became more frequent. This is why some of the clearest examples of natural selection in humans involve malaria. The sickle cell allele is the most famous. People who inherit one copy of certain sickle cell variants can receive partial protection against severe effects, while inheriting two copies can cause serious sickle cell disease. This evolutionary trade-off shows how powerful malaria selection can be. But the genetic relationship between humans and malaria is much larger than a single gene. Variants affecting red blood cells, immune signaling, cell surface receptors, inflammation, and other biological systems can all influence susceptibility. Burkina Faso has played an important role in this research because several ethno-linguistic populations live close enough to experience broadly similar malaria environments yet sometimes show different average responses to infection. Fulani and Mossi populations have been studied repeatedly in this context. Researchers observed that Fulani participants in some locations often showed lower parasite densities or distinctive immune responses compared with neighboring groups. Because these communities could live in the same general region, the difference raised an intriguing question. Was there something unusual in Fulani genetics that influenced resistance? Early investigations looked at well-known malaria protective variants but found that the observed Fulani advantage could not be explained simply by higher frequencies of classical mutations such as those already known from red blood cell disorders. This made the problem more complex. Later studies examined immune responses, gene expression, antibodies, and additional candidate variants. Some found differences between Fulani and Mossi participants in the way immune-related genes were activated. Others identified particular polymorphisms that might contribute to the pattern. Yet no single variant has provided a complete explanation. This is an important example of science remaining unfinished. A documentary can easily fall into the trap of saying, "Scientists discovered the Fulani malaria gene." The evidence does not justify that statement. The reality is more interesting. Malaria susceptibility is a complex trait influenced by many genetic and environmental factors. The immune system contains thousands of interacting components. Previous infections can alter future responses. Age matters. Nutrition matters. Local mosquito exposure matters. Housing conditions, seasonal rainfall, access to prevention, and medical treatment all matter. Genetics contributes to the pattern, but it does not operate alone. Mossi populations have also been central to large-scale malaria genomics. International research projects have included Mossi participants in genome-wide studies searching for variants associated with severe immunological challenges. These studies compare people who developed severe symptoms with individuals who were exposed but did not experience the same outcome. By scanning hundreds of thousands of genetic markers, researchers can identify regions of the genome where certain variants appear more or less frequently among specific cases. Whole genome sequencing from Mossi individuals has also helped build better African reference datasets. This matters because African genomes contain enormous diversity, and genetic patterns found in one region cannot always be accurately studied using reference data from another. For decades, global medical genomics relied disproportionately on European ancestry populations. That created a practical scientific problem. A genetic risk score developed using European participants might perform poorly in West Africans. A variant common among Mossi people might barely appear in a European dataset. Even the relationships between nearby genetic markers can differ because populations have distinct demographic histories. Expanding genomic research in Burkina Faso therefore improves more than knowledge of malaria. It helps medicine become more accurate for African populations. At the same time, malaria genetics teaches an important lesson about adaptation. Populations facing the same environmental pressures do not always evolve the same solution. Natural selection can only work with genetic variation already present or introduced through mutation and admixture. One population might possess a useful red blood cell variant. Another might develop a different immune response. Two communities living side by side can therefore show different average susceptibility without one being biologically stronger or weaker overall. Population-level differences should never be turned into stereotypes about individuals. A Fulani person is not automatically resistant to malaria. A Mossi person is not automatically more vulnerable. Genetic studies describe probabilities across groups, often under specific environmental conditions. Individuals overlap enormously. Clinical decisions must be based on the person, not an ethnic label. The same principle applies to every genetic pattern in this documentary. DNA can help reconstruct population history and biological adaptation, but it cannot rank people. What malaria adds to the Burkina Faso story is evidence that the genome is not merely a record of migration. It is also a record of environmental struggle. Different communities carried different ancestral variants into the region. Malaria then acted as a powerful filter, increasing some variants and reducing others. In another chapter, pastoral culture favored milk digestion. Here a severe pathogen shaped blood and immunity. Both forces demonstrate that human populations are constantly changing. But to understand where some of the ancestral variation originally came from, we must move much farther back in time, beyond modern ethnic identities and even beyond the modern landscape of northern Africa. Thousands of years ago, the Sahara was green, and that vanished environment may hold part of the explanation for connections visible across the Sahel today.
Today, the Sahara appears to be one of the greatest natural barriers on the planet. From a satellite image, enormous stretches of desert separate the Mediterranean coast from the savannas farther south. It is easy to imagine that northern and sub-Saharan Africa developed on opposite sides of an ancient wall, but the Sahara has not always looked like this. During the African humid period, particularly between roughly 11,000 and 5,000 years ago, large parts of the desert received far more rainfall than they do today. Lakes filled basins now covered in sand. Grasslands expanded. Rivers and wetlands created routes through areas that later became extremely dry. People lived in these landscapes, hunted animals, gathered plants, fished, and eventually herded livestock. The Sahara was not an empty desert separating populations. It was an inhabited ecological world. As climate changed, that world opened and closed repeatedly. Wetter conditions allowed people to move into regions previously difficult to occupy. Increasing aridity later pushed communities toward surviving water sources, the Nile Valley, North Africa, the Sahel, and other neighboring zones. These environmental changes likely played an important role in population history across the continent. Researchers investigating Fulani ancestry have therefore asked whether some of the genetic connections visible among modern Sahelian populations could preserve echoes of ancient Saharan interaction. Modern Fulani genomes show ancestry related mainly to West Africa, but also to North African and West Eurasian associated sources. One possibility is that part of this mixture developed through populations that lived across or near the Sahara during periods when movement was easier. But modern DNA can only provide indirect evidence. It tells scientists that populations mixed. Statistical methods can estimate approximate dates and identify likely genetic relatives. Yet without ancient genomes from the exact people involved, the reconstruction remains a model. For years, this was one of the biggest limitations in research on the Green Sahara. Archaeologists had discovered rock art, settlements, animal remains, pottery, and evidence of pastoralism, but ancient DNA preservation in hot African environments was extremely difficult. Then researchers recovered genome-wide DNA from ancient pastoral individuals buried at Takarkori in what is now southwestern Libya. These people lived around 7,000 years ago, when the Central Sahara was far greener than today. Their genomes produced a surprising result. Rather than resembling a simple mixture of sub-Saharan Africans and incoming Near Eastern pastoralists, they carried most of their ancestry from a deeply divergent North African lineage related to earlier North African hunter-gatherers. They showed only limited evidence of ancestry connected with populations from the eastern Mediterranean. This suggested that pastoralism in that part of the Sahara may have spread substantially through cultural transmission rather than requiring a massive migration of people from outside Africa. The discovery also demonstrated that the ancient Sahara contained genetically distinctive populations that are poorly represented by modern groups. This is crucial for interpreting Fulani ancestry. When genetic models describe part of Fulani ancestry as North African, the label does not refer to one timeless North African population. North Africa itself has changed repeatedly. Ancient indigenous lineages were later joined by ancestry from the Near East, Europe, and other African regions. A population living in North Africa 2,000 years ago could already carry a mixture created by many earlier migrations. If descendants of that population later moved south or contributed ancestry to Sahelian communities, they would bring all those earlier layers with them. A modern genetic algorithm might separate those layers into colors labeled North African, European-related, or West African, even though the historical migrants themselves belong to one community. This helps explain why ancestry percentages can oversimplify migration. People do not travel as separate genetic components. They travel as whole individuals whose genomes already contain the history of earlier mixtures. The Green Sahara also warns us against imagining one ancient Saharan people. The region covered millions of square kilometers. Communities living hundreds or thousands of kilometers apart could have different ancestry, technologies, diets, and languages. The two Takarkori individuals tell us about one ancient population in one part of the Central Sahara. They cannot represent everyone who lived across the region, nor can they be identified automatically as direct ancestors of modern Fulani or Burkina Faso populations. To establish a direct chain, researchers would need many more ancient genomes from different locations and periods, including samples closer to the modern Sahel and West Africa. That evidence does not yet exist in sufficient density. What we can say is that the Sahara repeatedly changed between barrier and corridor. Human populations moved through it and around it. North African populations themselves were genetically diverse. Pastoralism spread across these changing environments. Later, Sahelian populations inherited some of the consequences. The Fulani may preserve part of this complex history, but the exact routes are still being reconstructed. This uncertainty is important because it keeps the documentary anchored in evidence. The Green Sahara offers a plausible environmental framework. Ancient DNA provides new reference points, and modern genomes reveal later connections. But there are still missing populations between these pieces of evidence. Those missing ancestors form one of the greatest challenges in reconstructing the deep history of Burkina Faso and West Africa.
One reason the genetic history of Burkina Faso remains incomplete is simple. Ancient DNA survives poorly in many parts of West Africa. Heat, humidity, microbial activity, and soil chemistry can degrade genetic material inside bones and teeth. In cooler regions, scientists have sometimes recovered usable DNA from remains tens of thousands of years old. In tropical environments, preservation can be far more difficult. This has created a major imbalance in ancient genome research. Europe now has thousands of prehistoric genomes representing many regions and periods. Large parts of West Africa still have very few. For Burkina Faso, this means researchers often have to reconstruct ancient population history using modern DNA, archaeology, linguistics, oral traditions, and ancient genomes recovered from neighboring or more distant parts of Africa. Each type of evidence reveals something different. Archaeology can show when people occupied a settlement, produced iron, raised animals, or used particular pottery. Linguistics can reveal relationships among languages and sometimes suggest patterns of contact or expansion. Oral histories preserve memories of migration, kingdoms, political shifts, and descent, although they also change as they are passed through generations. Modern genetics can identify relationships among living populations and estimate past admixture. Ancient DNA has the unique advantage of directly sampling individuals who lived in earlier periods. Without it, scientists must often infer vanished populations indirectly. This problem has become obvious in other parts of Africa. Ancient genomes recovered from places such as Shumlaka in Cameroon revealed lineages and population relationships that were not obvious from modern DNA alone. The people buried there thousands of years ago did not fit simple assumptions based on modern populations living in the wider region. Their genomes showed that Western and Central African population structure was already complex deep in prehistory. Furthermore, recent large-scale sequencing efforts, such as high-coverage African genome datasets, have confirmed that deep, divergent lineages and complex introgression events are a fundamental part of West African genetic history, proving that human evolution here is far older and more intertwined than previous models suggested. The lesson for Burkina Faso is important. A modern population living in a particular place may not represent the people who occupied the same region thousands of years earlier. Communities can migrate, mix, disappear, or change identity. A modern reference group may be genetically related to an ancient population without being identical to it. This is why researchers use terms such as Yoruba-related ancestry or Mande-related ancestry carefully. Such labels usually mean that a modern population provides the closest available genetic comparison. They do not mean that the ancient population literally identified as Yoruba or Mande. The ancient ancestry may predate those modern identities by thousands of years. Similar caution is essential inside Burkina Faso. Mossi individuals cannot represent all Gur-speaking people. A small Bisa sample cannot capture the full diversity of every Bisa community. Fulani pastoralists near Ziniere cannot represent all Fulani populations across Africa. Even neighboring villages may differ because of founder effects or recent migration. Larger studies have begun to improve the picture. Researchers analyzing hundreds of men from more than 20 ethno-linguistic groups found very high paternal diversity across Burkina Faso. Autosomal forensic studies have also shown that many populations fall within a broad West African genetic cluster while retaining meaningful internal differences. But uneven sample sizes remain a limitation. One community may be represented by many participants, while another may have only a handful. A genetic graph can make both populations look equally precise even when the evidence behind them is not equally strong. Future research will need denser sampling within communities as well as broader sampling across the country. It will also need more ancient DNA, if preservation and ethical archaeological work make that possible. Yet more data alone are not enough. Genomic research also raises questions about ownership, consent, and interpretation. DNA samples contain information about individuals, families, and communities. Researchers must explain how samples will be stored, who can use the data, and whether future studies may ask questions beyond the original project. African genomics has sometimes suffered from a model in which samples were collected locally but the most important analysis, funding, and scientific leadership remained concentrated in institutions outside Africa. A stronger future requires researchers and institutions in Burkina Faso and elsewhere on the continent to lead projects, maintain data resources, train scientists, and determine which questions matter most to local communities. This is especially important because population genetics can be politically misused. A genetic study showing that two groups share ancestry cannot determine who has rights to land. A haplogroup cannot prove that one community is more authentic than another. Ancient Din cannot decide modern citizenship. Every living population is the product of earlier movement and mixture. Attempts to rank communities by who arrived first usually ignore how complicated human settlement really is. Even if hundreds of ancient genomes from Burkina Faso were discovered tomorrow, they would not replace history, archaeology, oral tradition, or culture. They would add another layer of evidence. The missing ancestors therefore represent both a scientific gap and a warning. We know enough to see that Burkina Faso contains deep and complicated population histories. We do not know enough to draw a complete genetic map from the early Holocene to the present. Some populations that contributed ancestry to people living there today may no longer exist as distinct groups. Others may be represented only indirectly in modern genomes. Future discoveries could confirm current models or change them dramatically. Good science must leave room for that possibility. And once we accept these limitations, we can finally return to the original question and ask what the available DNA truly allows us to say.
After following maternal lineages, paternal lineages, kingdoms, pastoral migrations, milk digestion, malaria, and the Green Sahara, we can return to the original question. What does the DNA of Burkina Faso's people actually tell us? The first answer is that there is no single Burkina Faso DNA. The country's populations belong broadly within the genetic landscape of West Africa, but they preserve significant internal diversity created by different histories of migration, marriage, geography, founder events, political expansion, and natural selection. Maternal DNA reveals one of the clearest examples. Across several Gur and Mande-speaking populations, mitochondrial lineages were highly diverse but did not divide strongly according to language. This suggests extensive female movement between communities over long periods. Marriage traditions, in which women move to the husband's residence, offer a strong explanation. Women carried mitochondrial lineages across social boundaries, while their children could grow up speaking a different community's language. The paternal story was different. Y chromosomes showed stronger structure related to language and geography. Some groups displayed unusually concentrated paternal lineages consistent with founder effects, bottlenecks, or historical expansion. Neighboring populations nevertheless exchanged male ancestry, showing that those boundaries were never absolute. The contrast between maternal and paternal DNA is therefore not a contradiction. It is evidence that men and women participated differently in population movement. Mossi genetic patterns provide one possible example of political expansion, leaving a sex-bias signature. A restricted set of paternal lineages may have expanded while women from many existing communities became incorporated into the growing population. But this does not mean the Mossi descend only from a small conquering group. Their overall ancestry is much broader. The Y chromosome represents only one paternal route among countless ancestors. Their mitochondrial diversity and wider genome point toward incorporation and mixture. Fulani populations reveal another kind of history. Genome-wide studies show predominantly West African ancestry, combined with varying amounts of ancestry connected through North Africa to West Eurasian-related sources. In Burkina Faso, a lactase persistence variant associated with adult milk digestion appears to have entered through this historical admixture and then increased dramatically because pastoral life made it useful. The result is a remarkable example of gene-culture coevolution. A cultural practice created an environment in which one genetic variant became advantageous.
Yet broader sampling across the Sahel proves that the Fulani themselves are genetically diverse. Communities in different regions mixed with different neighbors. Shared language and cultural identity did not require biological uniformity. Malaria adds another layer. Repeated exposure to one of humanity's most formidable health challenges created powerful selective pressure. Fulani and Mossi participants have sometimes shown different average responses to malaria, but researchers have not found one simple gene that explains everything. The evidence points toward complex interactions among multiple genetic variants, immune regulation, previous exposure, environment, and behavior. This reminds us that population differences are statistical patterns, not rigid biological rules. No individual's disease risk should be predicted only from ethnic identity. Deep history is even more uncertain. Mitochondrial data suggest ancient demographic expansion during a period of major climatic change. The Green Sahara created corridors where desert exists today. Ancient DNA from Saharan pastoralists has now shown that North Africa contained deeply differentiated populations before later migrations reshaped the region. Modern Fulani genomes may preserve some consequences of those ancient connections, but direct genetic links from specific prehistoric Saharan communities to modern Burkina Faso populations remain incomplete. More ancient DNA is needed. This uncertainty does not weaken the story. It makes the scientific process visible. Genetics works best when researchers distinguish what has been measured from what has been inferred. We can verify that certain populations share more genetic similarity than others. We can identify maternal and paternal structures. We can detect admixture and estimate its approximate timing. We can find variants shaped by strong natural selection, but we cannot use DNA to reconstruct every migration route with certainty, nor can we use it to define identity. Every person has far more genealogical ancestors than any one haplogroup can represent. A Y chromosome follows only one father-to-son path. Mitochondrial DNA follows only one mother-to-child path. Even autosomal DNA is reshuffled every generation, and some genuine genealogical ancestors eventually leave no identifiable genetic segment in a descendant. Genetic ancestry is therefore only part of human ancestry. Culture contains other forms of inheritance. People inherit languages, stories, beliefs, technologies, farming traditions, musical forms, religious practices, political institutions, and social relationships without these things being encoded in DNA. A genetic sequence cannot tell us which stories an ancestor told or what name they used for their community. That is why the strongest history of Burkina Faso comes from combining genetics with archaeology, linguistics, anthropology, oral history, and written records. When these sources are placed together, the country appears not as an isolated population but as part of a vast network connecting the savannas, the Sahel, North Africa, and other regions of West Africa. Its peoples were shaped by repeated contact. Women crossed community boundaries through marriage. Men established new paternal lineages or moved among neighboring settlements. Kingdoms expanded and incorporated existing populations. Pastoralists carried cattle and genes across long distances. Environmental pressures changed the frequencies of protective variants. Climate opened and closed migration routes. None of these processes created a biologically pure population, because purity is not how human history works. The deeper we look into genomes, the more mixture we find. The DNA of Burkina Faso, therefore, does not reveal one origin. It reveals many connections. And perhaps that is the most important finding of all. Human history is not preserved in separate genetic boxes. It is preserved in overlapping lineages, fragments of ancestry, adaptations, and relationships that continue to connect communities long after the events that created them have disappeared from memory. Burkina Faso's DNA does not tell the story of one people with one origin. It tells the story of many populations connected through thousands of years of movement, marriage, adaptation, and change. Maternal lineages crossed linguistic boundaries. Paternal lineages preserved other patterns of geography and social history. Mossi expansion may have concentrated particular male lines while incorporating diverse local ancestry. Fulani pastoralists carried genetic evidence of long-distance connections and a milk digestion adaptation strengthened by natural selection. Environmental pressures left another evolutionary mark across the region. And beyond all of these histories stands the changing Sahara, once greener, more populated, and far less of a barrier than it is today. Yet DNA can only preserve part of the past. It cannot define culture, identity, citizenship, or belonging. The strongest story emerges when genetics is placed beside archaeology, language, history, and human memory. And as new African genomes and ancient DNA are discovered, the story of Burkina Faso will continue to become deeper, more complex, and more connected.